but you can't see me.
Yeah.
Okay, I don't know what's going on with video.
How are you?
Good, thank you very much for the invitation. I hope things are going okay for you and the conference is going well. I've only been able to drop in for a few talks here and there.
It's my pleasure.
I might, Rodrigo, is it okay if I restart Zoom? If I don't have video, I mean, it's not critical, but it's always distracting.
No problem.
So I'm going to leave and come back in.
Você está me escutando, Rodrigo?
Tô sim, Thiago, tô sim.
Ah, deixa eu tentar aqui iniciar o vídeo. Ah, agora apareceu.
Tá, tô vendo sim. Acho que o João vai te promover agora para anfitrião, co-anfitrião, que você tem mais controles da sala.
Mas...
Professor Ayala, tudo bem?
Oi?
Professor Ayala também está na sala aqui.
O Ayala tá lá. Alex voltou.
Oh, strange. You can hear me still, but video is not working.
Can you try to share your screen?
Sure. All
It's
good?
fine.
It's good.
Hi, Tiago. How are you?
I'm fine, Alex. And you?
Not too bad. It has been too long since I visited.
Yes.
So things are well. Everyone is healthy, I hope, and safe.
Yeah, my friend. It's very difficult time to us, huh?
True, I think it is. Go ahead.
Oh, okay, you can talk, please.
Yeah. Yeah, I think it is difficult everywhere.
Well, we'll be better.
I think by the spring it will be much better.
I hope so.
I hope so also.
And the preparation to the holidays?
First, I have to grade exams, but then I will relax. But we will stay here. There is no travel really allowed in Alberta. How about you?
Okay. I will pass the holidays with my family in Cuiabá. Not travel.
Yep.
It's more safe to the children.
Yeah, I said you have two young ones now? 2 children you have now?
Yes, 2 children.
Yeah. That will make for
and a 6-year-old boy.
Well, the 6-year-old, it will be an exciting time of year. Yes, but my daughter will come home from university, so that will be nice.
It's good a time to see all with good health.
Yes, I think right now that is the most important thing. Although it is always disappointing to not visit more extended family, parents and grandparents and things. So
it's very difficult to all the family, to pass these holidays separated.
Yes. But work is still good?
Yes. Eu fui elevado aí, Rodrigo? Como é que tá o comando?
I think you
não
can start.
Tô vendo aqui, deixa eu ver aqui com o chat.
Do you want me to stop sharing my screen?
No, it's okay, Alex.
Okay.
You can start.
Posso começar a apresentação dele?
Uhum.
Good afternoon, everyone. It's a great honor to meet you, President Professor Alex Brown from Alberta, University of Alberta, Canada. Professor Brown has a great experience in theoretical and computational studies of molecular photochemistry and laser control, computational design of small molecules, photochemistry and the photophysics of fluorescent protein and their chromophores, understanding of novel bond and structural inorganic materials, molecular quantum dynamics, and optical control theory of laser control. Professor Alex, you have 1 hour to give your talk. You have a word.
Great. Well, thank you very much for the introduction, Tiago. And I'm sorry, but for some reason my video does not seem to be working. So you won't get to see me. Not that that's a bad thing. So I'd like to thank the organizers for the invitation to speak, and in particular, Rodrigo Lima for the invitation. And so I'm going to talk really a big overview of the work that we do on excited electronic states. And as you'll see by my affiliation, I'm in a department of chemistry, although I was trained as a physicist. But over my career, we've certainly moved closer and closer to doing chemistry problems. And since I know that much of the audience are physicists, I will try and not overwhelm you with chemistry, but try to give you a bigger picture of the types of problems that we can tackle using computational quantum chemistry approaches. And so really this is going to be more of a survey talk than some of the previous talks you've seen. And as has been mentioned at the end of several talks, if any questions arise that we can't address today, feel free to reach out to me by email, and my email address is here. So as Tiago alluded to, my group covers a diversity of problems in the realm of theoretical chemistry, really all the way from chemical physics to really true chemistry problems. And so we do work in quantum dynamics, in quantum control, although we've done less of that recently. We study multiphoton absorption in fluorescent proteins and their chromophores. We do a lot of different kinds of problems in excited electronic states, whether that's fundamental studies or specific problems where we work with experimentalists. And then we also study phosphorescent molecules. And we heard a little bit in the previous talk about the importance of spin orbit coupling, and I'll come back to that when I discuss these particular systems. And then we also do some main group chemistry, which is really can be connected to condensed matter physics. But really, today I want to try and talk you through 2 or maybe 3 stories about excited electronic states. And I'll start off by discussing phosphorescent molecules, why they're of interest, and what we can learn from computational studies. And then I'll talk through this idea of multiphoton absorption in fluorescent proteins, again, introducing the problem and what we can try to learn from computation and simulations. And then finally, if time permits, I'll give a brief discussion of some of our benchmark work on excited electronic states. So really testing methodologies for the best approach to compute excited electronic states. So I always feel it's important to start by acknowledging the people who do all the work. And since this is a survey study or survey talk where I'm going to discuss many of the projects we've undertaken, I really should thank my current group of graduate students and undergrads. The work I'll present will be a lot of the work of previous postdocs, graduate students, and quite a lot of work by undergraduate students in my group. But also, it would not be possible without a number of my collaborators, my colleagues, Eric Rivard and Robert Campbell, especially Professor Rivard. My collaborator in Brazil at the University of Mato Grosso, Professor Gabriel de Souza, and some recent work that we've done with the University of Tromsø and Dr. Milleke Felt, who was at Leuven in Belgium, but now is at the University of Münster in Germany. And of course, I should thank the people who give me money or computer time, either to fund these students directly or to fund the research that we do. And just for a little bit of fun, I thought I would contrast some of the beauty of Brazil. So these are photos on the left and right from a trip now many years ago to the Amazon with Professor de Souza and actually Professor Rivard, with what is more typical of Alberta, certainly at this point of time where we're in our winter. And I look forward to returning to Brazil in the not so distant future because it's been far too long. I think I've made 12 or 13 trips to Brazil and I look forward to coming back. So after thanking the people who've done all the work, I want to start with a sort of general overview of the way that we like to think, we meaning my research group or chemists in general, like to think about studying excited electronic states. And of course, this is a theorist view of electronic excitation. And so what we think about is we think about the ground state energy of a molecule as a function of the geometry. And of course, what we have is we have some particular geometry, which I denote by some collective variable R, that denotes all of the geometric parameters that describe the molecular geometry. There's some point where that's the most stable, the so-called equilibrium geometry. And so whenever we're doing a computational study, we have to find what this equilibrium geometry is. But of course, we're not interested necessarily in just the properties of this ground state molecule. The first thing to remember, and if you were at the previous talk, they talked about this in the condensed matter context where there are vibrations. So there's of course vibrations and therefore zero point vibrational energy in any molecular system. And so this is really the true ground state involves this 0 point energy. But our interest is in exciting to some excited electronic state. And so the easiest way to describe an excited electronic state is to simply compute it and determine what we call the vertical excitation energy. That is the energy of the excited electronic state at the geometry of the ground state. And this is what I've written down mathematically here, that this vertical excitation energy is the energy of the excited electronic state at the ground state geometry minus the energy of the ground state at the ground state geometry. But we can also determine some other interesting properties. So if we can actually optimize or find the most stable structure in the excited electronic state, which is also often very important, we can talk about the so-called adiabatic energy, which involves the difference in the energies between the ground state geometry and the optimized excited state geometry. We can also talk about the so-called fluorescence energy, which is just the difference between the excited state energy at the excited state geometry minus the ground state energy. Or we can talk about this so-called 0-0 excitation energy, which accounts for this 0 point vibrational motion. So if we want to study molecular problems like I'm going to discuss in the coming slides, we need to be able to have tools to compute both ground state geometries, excited electronic states, and to optimize geometries in those excited electronic states. And since this is a survey talk, I'm not going to talk about the details of these computational tools, save to say that there's been a lot of work over the last now more than 50 years in developing methodologies for computing excited electronic states of molecules. And of course, many of these methods have also been extended to condensed phases, so the solid state. But I'm again only going to focus on molecular systems. So one of the most common methods that I will talk about today and is widely used because of its computational efficiency and its applicability to a diversity of problems is so-called time-dependent density functional theory, or linear response time-dependent density functional theory. And this is a very common method that people use. But in addition to this density functional theory-based method, there are a lot of wave function-based methods based on solving or building on the Hartree-Fock approach in quantum chemistry. And these really fall into two main categories, so-called single reference methods. And these single reference methods, there's a long list of them of a variety of computational expense. So some of them are really intractable for some of the problems I'm going to talk about today, like this, for example, equation of motion coupled cluster method is really difficult computationally or impossible computationally to use for the large systems I'm going to talk about. You don't really need to know much about this alphabet soup of different methods, but just say there's a lot of them. There are also more complicated multi-reference approaches that are often needed, especially for a lot of the interesting problems that we want to study. And these are so-called not black box methods. You have to give some thought into how you're going to use them. There are also so-called semi-empirical approaches. There's a very old method, which is referred to as Zindo, and a method that's really become quite popular in both molecular systems and in the condensed phase, and that's time-dependent density functional tight binding theory, which is a built-on time-dependent density functional theory. So of course, this theory is not going to get you very far. What you actually need is computational tools that have implemented the theories. And we use a diversity of them in my lab, depending on the type of problem we want to solve. And so you can make a choice about what you want to use for these kind of problems. And the choice really depends on the type of problem you want to solve, because many of these approaches, the quantum chemistry methods, are only available in specific codes, while TD-DFT is available in many of them. Some of the wave function-based methods are only available in very specific chemistry programs. And of course, it also depends on your budget, because some of these software packages are quite expensive. And of course, the computational time you may need to solve the problem can also be expensive. So that really is not very much detail about the approaches we're going to use, but I really wanted to give you a basic understanding that we're using a variety of existing tools to tackle interesting problems. And so I would like to take some time to discuss those problems. And where I want to start is probably the problem that's most connected to condensed matter physics. And that's in the idea of light-emitting diodes. And LEDs are important because they provide energy-efficient lighting. And of course, what we desire is for these LEDs to emit in a diversity of different colors, red, blue, green. Because what you'd like to be able to do is combine them to get the whole spectrum of visible colors that we can see, and therefore we can use them for devices. And of course, the initial blue light-emitting diodes were based on gallium nitride, and the 2014 Nobel Prize in Physics was awarded to Akasaki, Yamano, and Nakamura for their invention of these efficient blue light-emitting diodes. But there are drawbacks for these solid-state LEDs, and that is their relatively low stability and short lifetimes, and the devices are difficult to fabricate. But of course, we know that they exist. And so one solution is organic light-emitting diodes. And we've already seen in the last year or so the emergence of actually commercial OLED devices. You can buy a television based on organic light-emitting diodes, or buy a computer monitor based on organic light-emitting diodes. But there are a lot of advantages of using organic light-emitting diode technology. And the real two big advantages are the tunability, because when you're using molecules or polymers to create your light-emitting diode, you have a wide diversity of chemical modification. Chemists can do magic with changing the properties and of systems by doing chemistry, changing chemical substituents. And we heard a bit about this in the previous talk in terms of the substitution of cobalt into your matrix to replace iridium. But imagine now where you can do it through covalent bonding of small or relatively large molecules. The other big advantage of OLEDs, or organic light-emitting diodes, is that their processability. Because if you're using polymers or molecules embedded in polymer matrices, you have solution phase chemistry, not solid state chemistry. And so this is often much more advantageous on the industrial scale when you're doing really liquid phase fabrication rather than solid state fabrication. So as I promised, there would not be too much chemistry, but here is a slide full of molecules, but there really is a take-home message. And so this was really work that came out of my colleague, Professor Rivard's lab. And they were synthesizing a number of chemical compounds with different substituents. In particular, they were looking at the substituents as you move down the periodic table from sulfur to selenium to tellurium, or perhaps to phosphorus, which is again up near sulfur, but is moving across now the periodic table. And their original interest in these class of compounds was to synthesize building blocks for polymer solar cells. That's what they wanted to do. There was no real interest in doing building OLEDs. But in the course of their synthesis, and here is one of the compounds. So this is a tellurium containing compound. It's connected to a couple so-called B-pin boron containing substituents. And so in synthesizing these compounds, what they saw is that if they had this in solution, so dissolved in some solvent like hexane, under UV light, they saw nothing. As they've later learned, there is really a weak emission from this, but it's very, very small. But what they found is when they crystallized this and put it into the solid state, under excitation by UV light, there was an extremely bright emission. And this is, of course, very interesting, where you go from something in the solution phase that does not emit to something in the solid state that emits very brightly. And so this was really serendipitous. And it also pointed to keeping your eyes open for interesting phenomena that you may see in the lab. But how can we explain this emission? So first, they did a series of experiments to try and understand how this emission emerged as you moved from the solution phase to the solid phase. And so what I have here on the figure shown is an emission spectrum of the system. So I have the wavelength of emission, and I have the intensity of the emission. And what you have is putting the system in a THF. This is the organic solvent where you saw no fluorescence. And then you're slowly adding water into that mixture. And what they saw is the emission started to grow. And of course, it's much more visible in this photograph of as we go from no water, so the purely organic solvent of THF, to adding water in a mole fraction of up to close to 100%. And what you see is the emission growing in intensity, which is really visible to the naked eye. So it's clear that the aggregation plays a critical role, but also the nature of the material plays an important role. So it only strongly emits in the aggregated state. And the quantum yield in the solid is around 15%, which is not bad, but it's not great. It's the quantum yield in solution is very, very low. As I said, it's not zero, but it's very small. So why do we get this phosphorescence? And I'm not going to describe the experiments that tell us why it's phosphorescence rather than fluorescence, but essentially with some collaborators in the Department of Engineering, we did a series of, or they did a series of time-resolved experiments showing that the time scale of emission is only, could correspond to the phosphorescence. It's a much longer time scale than one would expect if one has fluorescence in your system. So the first thing was we surmised, and it's been known for many other systems, that the emission in the solid state arises because you have a decreased rotational freedom of these two groups that are attached to your tellurium containing center. And so when you go into the solid state, these cannot rotate freely. And therefore you cannot easily dissipate energy in the excited electronic state. The only way you can dissipate that energy is not through this thermal or rotational motion, but through phosphorescence. The other thing that they looked at was changing this center. As I said, they looked at a number of these different heterocycles with different constituents besides tellurium. They also changed these groups that were attached to this unit. And so they considered a whole bunch of chemical modification where either they're changing the center, here you see a phosphorus, or here you see a selenium or a sulfur, or perhaps you had no center, like in this case here, or this case here. Or they changed the groups attached to this tellurium containing piece, where you say, for example, got rid of the B-pin groups and attached these furan rings. And what happened? Well, all of these were non-emissive. And so clearly, both the tellurium center and the groups attached played an important role in leading to the phosphorescence. So we need these. And initially, when we did this work, we thought you needed these B-pin groups, these boron containing ligands attached to this center, or these boron containing substituents. We've subsequently found that we actually don't need these. The Rivard group has done numerous other experiments, both with changing these groups, but also changing tellurium to the much more chemically benign compound of bismuth. And we've shown that you can still have phosphorescence when you go to a bismuth containing system, or if you change these substituents. However, you really need to be careful about what you do. And that's really where computation starts to play a role. It's understanding where this phosphorescence comes from. Why can you change this center? And why can you change these groups and still get phosphorescence? While in other cases, if you make these chemical modifications, you lose phosphorescence. And that really is the important role of computation, is understanding the mechanisms that lead to the emission that we see. Of course, if we understand the mechanisms of the emission, we could actually start to make predictions for new chemical syntheses that our colleagues should explore. So let me just give you a brief pictorial model of phosphorescence, and it builds really on that initial model that I had for emission and absorption in molecules. So again, you have a ground state of your molecule. Here it's a ground state singlet, and you have the minimum energy structure, and of course you have some vibrational 0-point energy on top of that electronic minimum energy. And we can obtain this by ground state density functional theory. And so everything I'm going to talk about here is with density functional theory. We can then compute an excited electronic state using time-dependent density functional theory. And so we can compute a vertical excitation energy. So this is our energy of absorption, and we're going to use the simplest model where it's the energy difference between the ground electronic state and the excited electronic state at the ground state geometry. So I'm just going to treat this as a stick. I'm going to get rid of sort of that dependence on geometry because I only have a single geometry of interest. But then if we want phosphorescence, we have to talk about spin-orbit coupling. And Eduardo Grande in the previous talk gave a really nice introduction to the importance of spin-orbit coupling as you move down the periodic table. And here with tellurium, we're looking at reasonably low down the periodic table. And so spin-orbit coupling, connecting singlet states with triplet states, is going to be important. And so we have some triplet state manifold that we can compute by time-dependent density functional theory. To understand how can we get spin-orbit coupling connecting a singlet to a triplet state, because these labels, singlet and triplet, are no longer good quantum numbers, and we have to talk about the quantum number J, just as Eduardo referred to in the previous talk to mine. So we have to talk about this spin-orbit coupling. And if we have this, we couple to some triplet state, and usually what we'll have is relaxation all the way back down to the lowest triplet state. And here we're going to find that using so-called unrestricted density functional theory, not time-dependent density functional theory. And then we can predict energies related to emission, and that emission is actually the phosphorescence energy that they measure experimentally. So we can get information about both the absorption and the phosphorescence through computational approaches. And we can also get this adiabatic energy or this 0-0 energy. And so how does it work? Well, if we look at the simplest compound where they first measured this phosphorescence, what we have is we have now experimentally computed using time-dependent density functional theory the excited state energies of all the singlet states, those are indicated by the letter S, and the triplet states, which are connected to phosphorescence. And these energies are all related to the ground electronic state energy. And what we have found is that there is a very energetically close or nearly degenerate triplet state connected to this singlet state that absorbs the light. If we now compute these energies using an approach where we have include the spin orbit coupling, we first have the singlet and triplet state energies. But now when we include spin orbit coupling, these labels are no longer valid. And we have to talk about the label of J, the total angular momentum, not the spin and orbital angular momentum. And what we find with this, including this spin orbit coupling, is that there is a strong mixing between these two states when we include spin orbit coupling. And in fact, you find that this state that absorbs is about 80% the singlet state and 20% T3. And it's this spin orbit coupling that leads to the connection between absorption and then the ability for the system to cross into the triplet manifold, relax down to this ground state triplet, and then emit the phosphorescence that we see, emit the photon related to the phosphorescence. So we have a really good handle computationally on the mechanism that leads to this phosphorescence. We can also easily use that model to explain what we see when we go across this substitution, which is moving down the periodic table from sulfur to selenium to tellurium, where these two systems, the sulfur and selenium containing system, are not phosphorescent. And we can get that solely by looking at the absorption energies. What we see for sulfur and for selenium is this triplet state that we need to access to get into the triplet manifold leading to phosphorescence is much, much higher in energy than we have for the tellurium containing system. So already we have an energetic penalty for trying to access this state. In fact, it's not energetically accessible. In addition, we're going to have much, much weaker spin orbit coupling because these are much lighter elements. And so we can explain this trend we see down the periodic table. Of course, we have to take care because there's known difficulties for TD-DFT and triplet states. And so one always has to know the limitations of the computational approaches that you're using to tackle specific problems. From these initial studies, what has been done both experimentally and computationally is they've moved on to how can we actually color tune this phosphorescence? How can we change from this bright green to something that's red or blue? And really they've only succeeded in moving towards the red. And they've done it by a diversity of chemical substitution. So here we have systems, similar to the original system that emitted in the green, but we have some systems that emit in the orange or yellow by changing the substituents around this tellurium center. The nice thing is we can explain this shifted emission computationally. So we have computed phosphorescence wavelengths. Here they are for the green emitting system and the orange emitting system. Experimentally, you see a shift of around 0.3 eV in the emission. And importantly, we can capture that computationally, and we can predict the trend in this emission as we change substituents. So in principle, we can make predictions for compounds that have yet to be synthesized. And I show this example, but we've studied quite a number of different systems in these tellurium containing cases looking at these phosphorescence wavelengths. And we have quite a reasonable model for predicting them. So this prediction is, I would say, excellent given the limitations of the method. So currently we have a number of things ongoing still in this area, which is trying to understand more quantitatively the role of spin orbit coupling. We are actually trying to compute and are successfully computing the the crossing points. So at what geometries do the singlet and triplet states cross? Because this gives us a better understanding of the mechanism that leads to the phosphorescence or the non-phosphorescence. There are limitations of DFT and TD-DFT, so we're looking at approaches that are not based on these approaches. So either a variety of wave function based methods, and we're trying to look at new compounds. So that's an example in the area of, I'll say, materials chemistry. But we also study problems that are much more at the interface of chemical biology. And so I'm going to totally change gears, but some of the tools I'm going to talk about, the computational tools, remain the same, but the problems we're tackling are completely different. So this is a very famous fluorescent protein, the green fluorescent protein. And fluorescent proteins are really important tools in chemical biology because they can be used to label a diversity of systems in a variety of in vivo and in vitro studies. And they can also be used to study mechanisms by creating fluorescent proteins that are responsive to the environment, whether that's the chemical environment, the pH environment, or changes in voltage if you want to do neuronal or brain imaging. And so these are really powerful tools for use in chemical biology. And because of the importance of these tools, the Nobel Prize in Chemistry was awarded in 2008 to 3 individuals associated with both the green fluorescent protein and the modifications of green fluorescent protein to turn it into a really, really useful tool in chemical biology. As I said, I promise not too much chemistry, but I think I have to talk about a little bit of chemistry. The main thing that makes green fluorescent protein interesting is because you don't have to introduce any chemicals or any external agent to make this a fluorescent protein, to make this a system that emits light. Rather, what happens is you have a folding of the protein, and I think all of us are familiar with protein folding in the general sense. But then once this protein is folded, 3 amino acid residues that exist come very close together, and they undergo some chemistry involving cyclization, dehydration, and oxidation to form a chromophore. And a chromophore is the part of this fluorescent protein that absorbs and emits light. And the nature of this chromophore depends on these 3 particular amino acids that come together to form the fluorescent protein. And that's really where the power of fluorescent proteins comes, is we can do modification of what these constituent amino acids are, in particular, this tyrosine 66. So by changing this tyrosine 66, we can change the color of both absorption and emission. And so over the past more than 30 years, a huge range of colors of fluorescent proteins have been derived, all the way from blue into the red. And they almost all, or many of them, involve changing this tyrosine 66. So you see that this part is different from this part, is different from this part, is different from this original green fluorescent protein. Sometimes the change is also in the charge state of the fluorescent protein chromophore. So here I have an anion. Sometimes that change is nearby residues that can interact with the protein. So here is so-called yellow fluorescent protein, where the chromophore is the same, but you have a nearby ring that pi stacks with the chromophore, again shifting the emission. Or sometimes you have this change in the backbone, which leads to a shift into the red of the emission. But there's lots of ways of tuning these fluorescent proteins. What we are particularly interested in fluorescent proteins is so-called multiphoton absorption. So everything I've drawn so far refers to one photon absorption. We absorb a single photon to go from a ground electronic state to an excited electronic state. But there's a great deal of interest in not this excitation where you lead to relaxation and then fluorescence, but excitation processes where you absorb simultaneously 2 photons of double the wavelength or half the energy. So you're still going from the same ground state to the same excited state. You're just doing it through the absorption of photons of double the wavelength. Or even 3 photon absorption, where you absorb 3 photons of triple the wavelength or 1/3 of the energy. These multiphoton absorption processes are useful because they provide better focus and they provide deeper penetration into thick tissue if you're trying to do in vivo imaging. And a lot of this comes about because of the absorption of water. So if you look at the absorption wavelengths in the visible from 400 to 700, the problem is you have hemoglobin. So you have blood or oxygenated blood that has strong absorptions in this visible region. So there was a lot of effort put into moving fluorescent proteins more and more into this red region. But a much easier way to do that is to not move to the red, but to take something that already exists at absorbing in the blue or the near UV and just double the wavelength. And then suddenly you're in this so-called window, the near infrared window between the strong absorption of hemoglobin and the strong absorption of water, where you can get the best optical properties for doing imaging. And so this is a challenging problem because you have both the intrinsic photophysical properties of the chromophore, that is the unit that absorbs and emits the light, but you also have the effects of this protein shell. And those effects are of 2 different types. It affects the conformation of the chromophore, that its geometry, and in particular how the geometry can change once it's excited. And you have the surrounding electric field or the electrostatics of the protein environment, which strongly impacts also the absorption and emission properties. So we've done a series of studies where we studied known fluorescent proteins, and here's just 9 of them shown here, of a variety of different absorption wavelengths. So they're all the way from red and orange to blue and the original green ones. And we studied these using time-dependent density functional theory. And we were looking at this 2 photon absorption. And we showed that time-dependent DFT for the isolated chromophores is a reasonable tool. There are weaknesses, but it predicts a lot of the trends in absorption that we see or are observed experimentally. But what we've been studying over the last 5 years is the idea that, well, if we're using the natural amino acids, for those of you who remember some of your basic high school or university biology, we only have 26 of them. But if we can diversify the number of amino acids that we can use by using so-called non-canonical amino acids, or moving beyond the 26 that we have in the natural occurring family, perhaps we can tune these photophysical properties of the multiphoton absorption and the emission even more. And so we've looked at a series of so-called green fluorescent protein-like chromophores and the so-called red fluorescent protein-like chromophores that have this extended structure that shifts the absorption to longer wavelengths. And we've done this for 50 models, which are built from a variety of available non-canonical amino acids. And we picked 10 which showed the largest 2 photon absorption. And so here are the 10. The identities of them are not critical, just to say that that they all have slightly different properties that lead to this increase in 2-photon absorption. But what's important is one of these is actually the gold fluorescent protein, which was one of the first fluorescent proteins made in the early 2000s, adopting the inclusion of a non-canonical amino acid. That is an amino acid moving beyond the 26 available amino acids. And so this gives us some, I would say, hope, some positive messaging that this approach of using non-canonical amino acids to tune photophysical properties will be important. So here's just some data. It's not so critical, just to show that for these 10, we see an increase in the 2-photon absorption cross-section. I think the important benchmark is we see that this goes from around 20 to 100. And for looking at the naturally occurring fluorescent proteins, the values are typically around 5 to 10. So we're getting almost an order of magnitude increase in the 2-photon absorption, which is critically important. These are two different models, and I'm not going to talk about the models they are. One of the things that we've been interested in is looking at computationally efficient approaches, because if we want to study these chromophores not isolated but inside the protein environment, we might need to study large numbers of chromophores, but also a large number of geometries where we sample them by molecular dynamics. The problem is time-dependent DFT is limited to around 50 atoms. And so we did some work looking at one of these semi-empirical approaches, so-called time-dependent density functional tight binding, to study this 2-photon absorption. And we've also been working hard to account for the environmental effects through quantum mechanical, molecular mechanical models. The environment's very much important for 2-photon absorption because of some experiments that have been done. So here are a series of experiments with the same chromophore. So that is the system that absorbs the light and emits the light does not change. All that changes for these so-called M-fruit fluorescent proteins is the protein environment. And what we see for the 2-photon absorption, which is given in red, is the protein environment can change this 2-photon cross-section by a factor of 6 or almost 10, while the corresponding 1-photon absorption is relatively flat. And so we need to take into account this environment. So the first part, we tried this semi-empirical approach. And the reason we tried it is because it's, here's some DFT results and the evaluation of TD-DFT takes on a CPU time of around 10,000 seconds. Here it's 10,000 times faster to do TD-DFTB. And so this is a very, very efficient approach for studying a large number of systems or a large number of geometries. And we've done this using a freely available code called DFTB+, and it's a great code for doing DFTB. The problem is we couldn't look at all of our systems because of some of their chemistry. And so we looked at 42 of 50 models. The problem is when we look at the comparison with quantum chemistry approaches, you see very large differences. Sometimes factors of 5, sometimes factors of 10, where blue is the TD-DFT and red is the DFTB. So it's clear, that this can't predict the magnitude or trend. So while it's useful for some problems, it's not useful for the problem we're interested in. What I want to do is I'm going to skip this. I'm cognizant of the time. I just want to take you to is this idea of looking at quantum mechanical, molecular mechanical methods. And this is where really I'm gonna end in the next 2 minutes. Because if we want to study this fluorescent proteins, we really need to study the full protein. And so we have to break the system into different levels of theory. One being, the chromophore itself, which we can study with the highest level of theory. Then the proteins surrounding that, which we're going to study usually using a molecular mechanical approach. Sorry, some issues here. And so we've done this with 2 different approaches, Unium, which is available in Gaussian. But then if we want to study 2-photon absorption, we've used the so-called polarized embedding model available in Dalton. And these are our most recent results that we have studying these fluorescent proteins, because we've studied gold FP. Sorry, let me just get through this here. Apologies. Where we've looked at the chromophore and the protein environment, as well as we often have to include some waters in the system. Sorry. Importantly, what we see, if I can get this right here, is that when we look at the isolated chromophore, when we look at different approaches, including the protein environment, we see differences anywhere from 20 to 100%. And so it's clear that we need to take into account this protein environment. So I know I went through this a bit quickly, but we've investigated multiphoton absorption for a variety of chromophores. And what we're really focused on is the QM-MM approaches to include protein effects. They really are critical to studying these systems. We've also been worried not just about the absorption, but about the fluorescence. We've also started extending our studies beyond 2-photon absorption to 3-photon absorption, and also to so-called non-degenerate 2-photon absorption. And we're also working on methods away from TD-DFT, and we're working with experimentalists. So where I want to go here, just 1 second. What's going on here? So where I want to go in my last slide is just the take-home messages. Since as I said, I would tell you 2 or 3 stories. The real take-home messages are quantum chemistry approaches are very useful to studying a diversity of physical problems involving excited electronic states. There are a lot of useful computational tools that one has access to study problems that you're interested in. But really, one of the things you must be careful about is to understand the strengths and limitations of the approach you choose. And that was really going to be the highlight of the 3rd problem, where we have studied a diversity of chemical approaches, or sorry, quantum chemistry approaches to study a problem, because we found that a number of them are insufficient to properly address the excited electronic states that we're interested in. So on that note, I would like to thank you for your time. I'm happy to take questions in the Q&A, and I'll end off by thanking the people who did all the work and the people who provide the funding. Thank you.
Thanks, Alex, for your time and share with us your knowledge. If you have a time to question. One question. Hello, Professor Brown. Thank you for talking. Have you on your team worked in turning non-fluorescent protein or fluorescent, especially lipoproteins such as lipase?
So we actually have not done any direct work looking at taking systems that are non-fluorescent and turning them fluorescent. I would say that the closest we get is some of our ongoing work is looking at systems that are weakly fluorescent and trying to turn them more fluorescent. I think that the challenge is that while we can predict quite accurately absorption, wavelengths, and also absorption cross sections, it's quite challenging to compute. While one can compute, I should say, fluorescent wavelengths, so the energies of fluorescence, computing sort of the quantum yield can be very challenging. However, you can, of course, get an idea of whether a system is perhaps going to be fluorescence by looking at its response to excitation. So looking at how the geometry changes in the excited electronic state. Because usually something that I didn't mention is most of these chromophores, so in the fluorescent proteins, are non-fluorescent when you take them out of the protein environment. And so really the protein environment is really critical for there to be any fluorescence. And that's often where you see this change from non-fluorescent to fluorescent behavior, is by more constraining the environment around the fluorescent protein, or the fluorescent chromophore itself.
Thanks, Alex, again. Have more questions?
Oh, so the limit of TD-DFT is 50 atoms. I mean, that's a ballpark figure. Of course, you can do larger systems with TD-DFT, but you can't go much bigger. So I mean, you know, you're not going to be able to simulate the thousands of atoms that we have in a fluorescent protein with TD-DFT. At least not, because again, if you're doing condensed matter where you can use periodic boundary conditions and you have small unit cells, then of course you can, in principle, go to very large systems. But in these molecular systems, we really struggle. So also depends on how big your computer is.
There is any more question? I think don't have more question. Thanks, Alex, for your speech.
Thank you. It was wonderful to be here.
And
as I said, if people have additional questions, do not hesitate to reach out by email.
Okay. I will pass your email to the students and the other colleagues. Thank you again, Alex.
Thank you.
Thank you. Our next invited, our next talk. Our next invited colleague is Professor Mauro Luciano Baesso. Professor Mauro, he's a full professor at Universidade Estadual de Maringá. He has a great experience in condensed matter physics with special attention to the development of special glass for generation of intelligence, white light, and for the construction of new laser for biological application, and photothermal phenomena applying the study of special glass, biological systems, biomaterials, and photopharmaceuticals. Professor Mauro, you have a half hour to your talk.
Oh, thank you. I have to share my slide, is it?
Yes, please.
Can you see there the title of the presentation?
Yes, Professor.
Well, first of all, I'd like to thank you, the organizer, for the invitation, and especially Professor Rodrigo Lima. It's a pleasure to talk to you. And probably the title of my presentation is a little bit, I'd say, optimist, because nowadays there are a lot of efforts trying to use photothermal methods to as a tools for preclinical or for clinical and imaging diagnosis. No one of these techniques are available yet to be used as a regular technique, but there are many possible advantages that have been said that could bring for a new knowledge in terms of earlier detection of any kind of disease. So for those that doesn't know Maringá and my institution, we are in south of Brazil, in the province of Paraná, in the north of Paraná. My institution has about 50 years old now. There is some data here regarding the number of students and graduate, undergraduate, and we have a hospital and some experimental farms, just for you to know about that. Well, along with the last 30 years, we have things that happen because we have worked in a multidisciplinary way. So as you see here, we are working with dentistry department, biology, pharmacy, and also some kind of research, and also other materials like optical glass for laser and something like that. As a multidisciplinary work, especially in the health science area, in the physics department, we do the measurement with the mainly phototherm and complementary method, like Raman spectroscopy, FTIR, conventional spectroscopy, and of course, all the electronic techniques also for the characterization. The idea is to try to do physical-chemical analysis of the disease, of the tissue, in vivo, in vitro, and ex vivo, and when it's animal, and this is the way we work. Well, just to give a message today, this image is an arrangement. It's not from our laboratory, from the Professor Hazansky from the Federal Institute of Zurich. I've been there in the beginning of this year. And the imaging using photoacoustic detection, ultrasound photoacoustic detection, is almost there for extensive applications. And as you can see here, there is a lot of challenge in that, because when you have an ultrasound generated by laser, for example, the sample like in this position, or like it is positioned here, you can see. When you have lasers around in this detection and detection system, and this laser can induce an ultrasound in some position inside in this case the finger, because we use a wavelength of the laser which is transparent in this area, around 600, 700, 800 nanometers. And the ultrasound is generated, and then it's captured by the sensor, which is usually 10 centimeters away. So there is all the imaging process, this inverse problem, and to get this image and reconstruct the image in real time and for in vivo measurement. This is we are still we are not still doing that, but the idea is because the challenge that it should be, it's my view that the challenge in this area is first to get imaging. The people are already doing that, but the challenge is to get, for example, if you want to differentiate between malignant and not malignant cancer, probably it's necessary for you to use excitation where you have functional group sensitivity for the absorption, like in the infrared. In the mid-infrared, you can see, for example, the change in DNA, which is some kind of fingerprint for malignant cancer, for example. So today, this measurement is not available yet, but the lasers is already available in this area, so quantum cascade laser. So this is maybe a future for that. Just for you to get an idea, this comment is on Nature on the beginning of this year regarding that the imaging with photoacoustic ultrasound is already available. This is of course in animals. So just to get a message for you that the student that should think for the future to solve this big problem, which is a discrimination between malignant and non-malignant cancer. Today, although we have a lot of techniques, but the pathologist will be the final answer for the differentiation. Of course, if you would have a technique that could help on that, it should be very important for the area. But for you just to have an idea, it's a image from Nature Photonics paper from the people from Caltech. You see the measurement, the measurements are with animals. You can see like that, but you have to have a transducer between the sample and the detector, and then you can have images like that, which is already published in the literature. But for you, already seen, mid-infrared is already being tried. People are already trying, and this work is also from this year with the people from Munich, and they are already getting measurement in the mid-infrared, trying to do what I said before. And also, this is all their imaging when they. And I just showed this their transparency to show this spectrum, because the important thing is to get, if you are able to get imaging with wavelength in the infrared between in the mid-infrared, that you can see DNA change due to malignant cancer. So this is in cell culture that's already done. And just now I'm going to talk about we're going to show some work we have done here, mainly looking at the physical chemical change in biological tissue and how the photoacoustic and photothermal techniques and also other Raman spectroscopy can be used together, trying to give some answer for physical chemical interaction between changing tissue by some disease or even for characterization of tissues. Of course, I'm going to focus on infrared spectroscopy, especially in the mid-infrared, because you can see the vibration and rotation of the molecules, and then the functional groups, which are very important for living cells. And also, sometimes we do some visible spectroscopy too. Okay. How we localize what we are doing? So you see the energy diagram. For us in the photothermal area, all the excitation that you can produce in your sample, at the end what you have, the energy going to ground state, you get heat generation, and that is what we detect in our measurements. So when you use photothermal techniques, you detect the heat generated by the sample. And then from there, you can do complementary measurements like using luminescence, fluorescence, or even Raman spectroscopy, depending on the material you want to study. Just to localize, there are a lot of photothermal techniques, but the focus would be actually in the pressure technique that we use, like photoacoustic spectroscopy in the visible and also in the medium infrared. But also now this challenge we want to do is ultrasound, as I said, looking at imaging, which I think will be the future for our area. I mean, I think the imaging research will be important for the photothermal area in the next future. And also a technique that we work here, which is photothermal lens and photothermal mirror technique. I'm going to show why this technique, I think, could be used for imaging also, to complement what we measure with ultrasound techniques. Okay, just for those that doesn't have too much experience, I'm going to use like a conventional sorry for that. Using a conventional photoacoustic spectroscopy, you have a modulated light inside in a sample in a closed cell, and then you have the absorption of the light. If the light is absorbed here, now you can see here, if the light is absorbed, and then part of the energy can be transferred to heat, and the heat generated will be captured by a microphone that is positioned here, and then you synchronize with a modulation frequency, and then you can get the photoacoustic signal. So you get the heat generated after absorption of the light. One important point is in this technique that when you change the modulation frequency of your chopper, you can get different depth penetration of your sample, and then you can do some kind of depth profile imaging or for your material. And also, this is for when you use UV spectroscopy. This is a conventional one. You use a xenon lamp and with a monochromator, and then you have a measurement wavelength by wavelength. But of course, there are commercial equipment today that you can use FTIR detection, like this cantilever microphone on the Gazero cell, and then you can do the whole spectrum, as you know, in the infrared by FTIR, and then you can also measure the absorption coefficients of the samples in the mid-infrared and near-infrared. The idea is, of course, when you go to the mid-infrared, as I said before, look at the functional groups of the biological tissue and how it changes with some disease or any changes you try to induce in the tissue. Okay, so this is the two photothermal spectroscopy that's photoacoustic that we can have. You have no commercial equipment in the UV visible. This is homemade, but you have commercial one for the infrared. I'm going to show some results which I think it's very important. First is it's a subject that is important for us in South America, because this fungus, Fusarium, is very prevalent in our region, and it can cause a lot of problem in disease. How we did the measurement? We did a set of experiments. The first was the metabolites of the fungus was applied in the skin of a mouse, rats, and then we say it ex vivo measurement. What it means? You apply the fungus in the skin surface, and then you have a protocol with as a function of time that you, of course, you make the euthanasia of the rats, and then from time to time do the measurements to see if the fungus was able to get the penetration in the dermis, for example, of the animals, and what kind of problem it can cause in the skin. And of course, in the measurement, we applied in the skin, and then we move back, and then we illuminate the sample on the other side, and then we try to detect the presence of the fungus, and also the changes in the skin that they could induce. For example, here, what kind of, you go to the infrared, you look at the absorption bands related with some kind of, for example, collagens. What we observed in this work was the fungus metabolic was reached the dermal region and then was able to disorganize the collagen of the skin and then induce the apoptosis in the cells. So one important point when you do also with when you use rats for this kind of measurement, they are immunocompetent. They can recover from after some days of occupation of the fungus. Other measurement was instead of applying the fungus on the metabolites on the skin, it was injected in the caudal of the animal, and then we, after protocols of some days, for example, in this way, we start with the spline, look at the spline, the chains. You see what kind of measurement you do. Absorption bands, structure of absorption bands, and then you do the core fitting, and then you, in this case, it was 8 weeks of measurements. So you have animals distribute along the 8 weeks, and then to see how the absorption bands happen. In the beginning, we did the core fitting, but nowadays we are using PCA statistical treatment, and then at least for a macro evaluation. And then after that, looking at the absorption band, how they change during the time, and then you can look at what happened in your results. The same results in lungs. No? We did also the measurements, and this work was already published also. And then this is the results. So you can see we do the measurements as a function of time, and then you evaluate how the animals change and then evolve. Of course, it's very important to say this kind of study, it's necessary all the time. You have biological people from biology that do. For example, histology is very important for this kind of measurement. You have to compare the spectroscopic data you do with the data that people get from the biology, and then to try to analyze what is going on in the tissue with time. This study was done with Raman spectroscopy, which gave the similar spectrum, different kind of detection. And then we look at the liver. I'm showing it to you because we observe very special thing that you see in the histology data. It looks like the body is exposing the fungus from the liver, from the animals, and then we evaluate the bands from the evolution of the bands. You can see here that the animals, after, in this case, for 8 weeks also, you can see the data. The animal recover the structure of the bands with time. This paper is published in +1. Now I'm going to change to dentistry area, because we are also doing we have done a lot of work in dentistry also. The first one, it's a combination of photoacoustic spectroscopy with Raman spectroscopy to evaluate the effect that hydrogen peroxide gel induce in the tooth when they are whitening. So we applied the substance here, as it's shown here, in the dentin, and then we try to detect its. We applied in the enamel, and then try to detect the gel in the dentin, which is in the interface between the pulp and the tooth. We did that, so you see the evolution of the bands detected by Raman spectroscopy as a function of time. In fact, it's that way, in a function of time. And then how the absorption bands change with time in photoacoustic spectroscopy. Each absorption band, you can see the organic structure of the tooth is changing, is being disrupted by the hydrogen peroxide, and in the same time we were able to we scanned the tools with the Raman spectroscopy, and it was possible to see that all the hydrogen peroxide stay in the great concentration between the enamel and the dentin, and which is and propagates through the dentin slowly when you have this application. That's why it's very important for the people from the industries, because about 30% of the hydrogen peroxide that was applied reach the pulp, where that's why you feel pain when this kind of substance is used. Another study was with interest in dentin for photodynamic therapy. We made the profile when the people apply some dyes in the tools, and then after the dyes, we measured with photoacoustic spectroscopy the penetration of the dyes through the teeth, right? And then we have the concentration. It is a parameter that's important for the people from dentistry, for they to evaluate the amount of dyes to use and the intensity of the light that could be used. And this is a very simple experiment using UVVs of photoacoustic spectroscopy with this setup, which is homemade here. This is the result. We made a map for the penetration of the dye through this. And in the last one, it's not a photothermal technique. We use a thermographic camera. Sometimes it's important to get the temperature of the tissue, and we evaluate how it changes when you use we preparate the cement from during the preparation for tools restoration. This is a very important process, because the time of necessary to get the cement red, it's very important for the kind of interaction, chemical bond that you make between the cement and the tools, the link between those. And we use a thermographic camera. You put for example, here, and you get imaging it's very fast measurement. Every 20 seconds it's possible to measure it. I'm going to show you here, as for an idea. The first 20 seconds you have this band, and then after this is measured with HER, and then I will show the camera. After 1,800 seconds, you see the absorption bands, how they change a lot of the intensity, and we monitor the change in the absorption bands, and then we got the image with the thermographic camera. It was very nice, because some cements, depending on the origin of them, they have two stages. Of first, it hits up. It's the first 20 seconds you cannot measure before that. It should be very fast. And then you have the minimum, and then the temperature goes up again, like this minimum, it goes up again. You have two stages of reaction between the cement, and then you see the variation of temperatures detected, and we correlate the changes in the chemical bonds in the cement with the pattern of temperature to understand how it happened in terms of the reaction, the hydrolytic reaction in the tools. Well, I think I have a short time, and well, just to say, because the technique we want to do imaging, because I wanted to finish with imaging. I start to talk about that. And this technique, which we call pass a laser beam in a material, we developed here. I can just show the slides where we have this technique here. It's called thermal mirror technique. You inside the laser in the sample, you get some kind of change in the surface, and you can measure the change in surface. And just to show you, we have all the model, we can calculate the intensity of the laser after reflection, and then you can get quantitative measurements. We applied that in this work in water, just to get collision of a photon in the surface of water, and the change in the surface was we made the model, we evaluated the moment of the photon, and just to show you that technique is in our view ready for to be used for imaging measurements. This is just pulsed laser. So the idea is to use that technique, the thermal mirror technique, to change the position of the sample, and then get imaging surface, and then try to couple with the the challenge that we say imaging to get imaging from excitation in this region. Why? As I said before in the beginning of the presentation, because in this spectral region, we have a change in DNA, which is the important information to say that you have, for example, a malignant cancer in the tissue you want to analyze. Well, I'm sorry, maybe I think I passed a little bit of my time. And I want to thank you, all the people. We have a big group in the physics department. We have about 35 persons with students from all levels. And we are in seven professors and lecturers, which are involved in this multidisciplinary research. Of course, I'm not counting the people from biology, pharmacy, dentistry, and these are some kind of multidisciplinary work we try to do. And then we'd like to thank you, our agents that help with financial support. And then I thank you for the opportunity to present this work.
Thank you, Professor Mauro, for your lecture, for your time, for your knowledge. There is any question to Professor Mauro?
Should I see the chat?
No question.
Okay.
No question, Professor Mauro. Thanks
okay.
Again.
Okay, thank you very much.
Professor, our next guest is Professor Alejandro Ayala. Professor Ayala, are you there?
Hey, hello, I'm here. Just let me turn on my camera. Hello, everyone.
Hello, Professor Ayala. How are you?
Fine. Thank you very much for inviting me to participate of this meeting. Let me just put my screen.
Your resume. Professor Ayala had a great experience with the Raman spectroscopy. He studied organic and inorganic crystals, have a great experience in high pressure study. He's an associate professor at Physics Department of Federal University of Ceará. Professor Ayala, you have a half hour to give your lecture.
Okay. Okay. I think are you seeing my presentation?
Yes.
Okay. Okay. First of all, thank you very much again for inviting me to show what we are doing in our university. Thiago told you I am professor of the physics department of the Federal University of Ceará. We have a long tradition on Raman spectroscopy. It's maybe from the 70s that we are doing Raman spectroscopy there. And recently we also starting working or we install a crystallography laboratory for single crystal X-ray diffraction. And what I will show you is some of the results we got in combining Raman spectroscopy and X-ray diffraction. We have installed a single crystal diffractometer. At this moment, it's the only one working in the Brazilian north and northeast region. It's a multi-story facility, and everyone who needs it will contact us. We will be glad to receive collaboration or receive samples in our lab. But we also, for this work, we also have used the Brazilian Synchron facility, especially the XDS beam line, which now this facility is disabled, but it will resurge in the series maybe next year as the EMA beam line, which is the extremes conditions beam line. And we also are doing in lab Raman spectroscopy measurements in our equipment, where we are able to measure under different conditions, especially temperature and pressure, which I will use in my talk. But since there are a lot of students in the audience, I will ask your permission to the most experienced people to give me time to use just 3 slides to talk a little bit about the techniques we are using. And very quickly, the first one is X-ray diffraction. Both techniques are very old, are from the first half of the 20th century. But this time, people realize that they can get information of the regular arrangement of atoms inside of a crystal using a diffraction experiment like the one you study in optics, just to use the periodicity of the atoms to produce this constructive interference. The first experimental result was from von Laue, but the Braggs, father and son, they developed a theory to correlate the experimental results with the position of the atoms. And maybe you have listened in different contexts about the Bragg law, which correlate the position of the distance between the atoms with the condition for the constructive interference. So we will measure all the direction where we will have constructive interference. And in the case of single crystal X-ray diffraction, we will get all these different directions. From this experimental result, we will get the intensity of all direction where we have a constructive interference. And from this information, we can recover the density of the electronic density, and from the electronic density, we will get the crystalline structure. That is how the atoms are arranged inside of the crystal. On the other hand, we can use powder X-ray diffraction. In this case, we have a lot of tiny crystal randomly oriented, and so instead of have an image like that, we will have something like all the contribution of all the orientation, and at the end we will have, instead of different well-defined dots, we will have some circles. And from usually we get just one direction here, and from there we have intensity as a function of 2 theta, and from there we can use or the Rietveld method from determine the crystalline structure, or Levi-Paul's method, just to get some information about lattice parameters or things like that. So on the other hand, let's say in this case, we are getting information of the average structure in the whole crystal. On the other hand, we can just try to measure how the electromagnetic radiation interact with the molecules. For simplicity, we will use just a single molecule, but in our case, we will study crystal. And the vibration of the molecules, there are quantified, and there are vibrational energy levels. And what we can use is if we send a photon exactly with the energy between those levels, this photon will be absorbed. This process is known as the infrared absorption, and we used to do infrared spectroscopy, which is usually infrared absorption spectroscopy. But in our case, we are more interested in Raman spectroscopy. Raman spectroscopy is like where in this case, a scattering of the light. The photon will arrive to the molecule, and it will induce these vibrations, but it will transfer a little bit energy just to produce this vibration, and it will follow with a large amount of energy. So we will use a photon with energy larger than the one of the vibrational transition, and we will lose, the photon will be absorbed in some kind of vibration, virtual states, and will be emitted with less energy. The difference will be the vibrational spectrum. The photon we can also get energy from a vibration, and to lower the vibrational energy level. But since these ones are the more efficient, the most intense, which are this one, we will study just this part of the vibrational spectrum, which is called the Stokes-Raman spectrum. If you compare Raman with X-ray diffraction, both of them are getting information about the crystalline structure, because these vibrations, molecular vibration we are discussing here, in the case of the single molecule, in the case of the crystal are the phonons. They are whole vibration in the whole crystal. So in one hand, by X-ray diffraction get information of the average structure. On the other hand, vibrational spectroscopy, especially Raman, is getting information more locally around the molecules or around the atoms which are vibrating. So this it's a very good combination, because we are looking at the crystal in two scales: as a global, as an average, and locally. What we will study are called hybrid halide perovskites. Hybrid because they can be purely inorganic, or they also can have here an organic cation. But beside this organic and anorganic cation here, we will have an halide octaedra with a metal inside, usually, for example, lead, germanium, tin, several of them, and we will have an halide on the corner. These are very old compounds. They are described first at the end of the 19th century. The first hybrid perovskite is the end of the 70s. But the most important, one of the most relevant results is from the end of the first decade of the 21st century, where it was proved that the organic inorganic perovskite can work as a solar cell. It was a quite poor solar cell. The solar cell was a quite poor efficiency, 3%, but with the future, with further development, they reach around 22% around 2000, 20% around 2015, and with that, the research in the field really exploded because they are we are reaching now near to 30% of efficient with a light perovskite, which is above the traditional solar cell. It has an advantage. They can be produced very easily, but for example, by solvent method, which is much easier than to produce a semiconductor solar cell. On the other hand, the problem they have is they are we still need to solve some instability problems because they are very sensitive to humidity, light, temperature. And the most efficient one used to be based on LED. And you know that LED is not ecologically friendly, and there is a lot of work to get rid of the LED. The title of my talk is about dimensionality, but what means dimensionality in this set? What is well known is the halide octaedra is the most important entity of these materials. So in the classic perovskite structure, we have a 3D network of octaedra. In those called 2D octaedra, then we have planes of octaedra. Be careful with that because these are not perovskites anymore. They are perovskites related, but usually people call them 2D perovskite, but this is not a correct description of the compound, but you will see things like that in all the literature. When the octaedra forms columns, we have a 1T. When the octaedras are isolated, we have a 0D. We can produce those things using several methods. I will not focus in the synthesis at this moment, but we can produce their very nice crystal with cubics or other crystalline habits, and they are, I would say, quite easy to produce. So I will start talking about my first example as a very classic Raman spectroscopy study in material science, which is to study structural phase transition. So in this perovskite, which is a LED-free perovskite, we have a so-called double perovskite structure because the two atoms inside of the halide octaedra are intercalated. And what X-ray diffraction and showed is there is a structural phase transition around 122 Kelvin. The photoluminescence also, or the absorbance of the light also shows a change at this point. And if we calculate the Raman spectra, we can associate the different bands to the different kind of movements of the cations and anions in the compound. If we do some Raman spectroscopy, we can start Raman spectroscopy as a function of the temperature. This reproduces quite, let's say, the theory reproduces quite well the experimental results. And you can see that is if you take a first look to the spectra, it looks like nothing happens. But measuring, taking the position of the Raman peak as a function of the temperature, we are using, if you check here, we are using a unit, is the very common unit for vibration spectroscopy, which is called wave number, the inverse of the centimeter. But it is proportional to energy. So higher wave numbers are higher energy. It's a traditional unit of the field. And you can see that, in fact, we really have a change in the behavior of the phonons. The phonons, remember, they are associated with the structure, and these phonons are this kind of behavior is very well known in vibrational spectroscopy, which is called soft mode, because the mode is becoming soft when you go near to the phase transition. And this kind of transition sometimes is associated to a ferroelastic transition, which is I can use where we can have different states of stress in the inside of the crystal. And if we take a look at how the birefringent of the crystal change as a function of the temperature, we really see, sorry, this is too unswitched, but between around 120 and we can see a change in the color of the crystal. Or let's say it's a change in the birefringent, because we are more looking to the crystal between crossed polarized, and which is associated to the phase transition. Another problem I would like to show you is now is a 2D perovskite, where we have planes of octaedra, which are lead octaedra. The octaedra are the lead iodine chloride. You can see how the iodine and the chloride are distributed in those octaedra. And this is interesting because it has a nice photoluminescence. And photoluminescence is a very important property for this material. And you can see that the photoluminescence is changing as a function of the temperature. But in principle, no phase transition can be observed in this material. With this theoretical calculation, again, you can see how the vibration is a material, and those reproducing quite well those of the observed experimentally. But there is one here which is not predicted in the calculation. And this and very weak here can be associated to something called overtones. In principle, if the crystal is purely harmonic, overtones are not allowed. It is due to the quantum selection rule of the transition of the harmonic oscillation. Maybe you maybe you study something like that in the quantum mechanics course. But if there is something inducing some anharmonicity, you can see several some overtones, which can be 2 times something. They could be a combination of 2, or they could also be some vibrational modes which are just allowed for symmetry to be observed in the infrared, but due to this anharmonicity, you can observe those in the Raman spectra also. That there are some vibrational modes which can be observed in the Raman spectra. Some of them can be observed in the infrared, some of them in both, and those things are determined by the symmetry of the crystal. In this case, which the modes observed in Raman are not allowed to be observed in the infrared and vice versa. But the harmonicity could break this rule. If we measure as a function of low temperature, you can see that harmonicity is growing at low temperature. And there are some strange things happening around 100 Kelvin, but no phase transition can be observed. And when we study the luminescence, which is the, let's say, the most important property maybe here, you can see what it was previously reported. The luminescence is increasing up to liquid nitrogen, but when we go below liquid nitrogen, new bands appear much more stronger than the original one. And when we investigate how these bonds change as a function of the temperature, we can describe this behavior as a exciton-phonon coupling. But if you have an exciton-phonon coupling, the phonon involved could not be a Raman band, but it must be an infrared band. And from the feed, we calculate the band should be the band should you have more or less 150 wave number. And if we go to the quantum DFT calculation, we can see that in fact there is a infrared band mode which we have around 150 wave number, and which is the one associated to the phase transition, to the electron phonon coupling. So quickly, just a last example, we have been working in these materials which have this 5 diagram, but I will discuss quickly this one, which is a cesium lead bromine or iodine. In fact, we were able to do some doping of the bromine with iodine, and you can see here first that with chlorine this bond is a high wave number. But remember, in the harmonic oscillator, the frequency of the oscillator is inversely proportional to the mass. When you increase the mass, the frequency is reduced. Since frequency is proportional to energy or wave number, the wave number is reduced. When you increase even more, introducing iodine, it is reduced even more. The cesium and iodine are completely distributed in the sample, but when we determine by single crystal electrolyte diffraction the structure, you can see that the iodine is only localized on this part of the structure. It is not substituting these atoms. Okay. If we study Raman spectroscopy, and now we are doing Raman spectroscopy under pressure. To do pressure, the sample is a tiny sample is placed between two diamonds with a liquid just to induce hydrostatic pressure. And these diamonds, they are forced to go one towards the other, and in order to reach very, very high pressure. Okay, maybe you listen presently of the room temperature superconductor, which are produced at very high pressure, but they are even much higher than the one we are using. But in any case, we are doing pressure. If you see the Raman spectra, you can see how the Raman spectra is changing as a function of the pressure. And an interesting thing is when we release the pressure, the structure is recovered. So it is an irreversible transition. And when we study the phonons, you can see how the phonons are changing, suggesting that there is a couple of phase transitions and one CPA and around 4.3 CPA. Looking to the luminescence, it is very interesting. You are seeing the luminescence increase and later it decrease and go to zero. So the pressure is quenching is killing the luminescence, but it disappears before the second phase transition. So this process is not directly associated to a structural phase transition. We studied this in the synchrotron when we measure X-ray diffraction as a function of pressure, and we were able to determine the crystalline structure using Rydberg. And we can see, we have confirmed with X-ray diffraction the structural phase transition, but we are interested in trying to understand how the structure is related to the luminescence. And when you study how these octahedras change with the pressure, we can see that in the first transition, there is a change in the distant bromine iodide and the lead and the bromine iodide distance, and in the distant in the lead bromine angle. And let's say in the distant and this distance here. This distance is changing, and this angle is also changing. There is inside of this structural phase, there is no structural phase transition, but there is a change here. And when we compare this with the photoluminescence, you can see that in fact, when the photoluminescence is doing a change, there is in fact a change in the bromine octahedra. So the change, you can see that the change in the luminescence are directly related, and how the octahedra change. This change will change the energy level of the excitons, which lies inside of this octahedra, and these excitons are those which produce the luminescence. So I will go directly to the end of my talk with that. So as a final remarks, we can see that it's very common that besides on this material there are a lot of structural. It is very common to see low pressure structural phase transition. It is also very common to see a change in the luminescence, which is the luminescence is suppressed a little bit above the first structural phase transition, but not at another structural phase transition. There is a red shift followed by a blue shift in these compounds around, and this change happens around, not exactly, but around the phase transition. And also the phase transitions are always associated to distortions of the halide octahedra, which are always reversible. And Raman and the vibration spectra and the photoluminescence spectra are strongly related because the exciton phonon coupling plays a very important role in the photoluminescence emission. And with that, I would like to thank all the agencies which are supporting our research, the Sirius, the LMS and Sirius facility, which are extremely important for our work. University Professor Ricardo Baesso from the University of Uruguay, which support us with the calculation, and to this research group, which is a very multidisciplinary, involving a lot of people from physics, pharmacy, chemistry, where we are developing a lot of work on solid state physics and also on pharmaceutical application of solid state physics. And with that, thank you very much, and we'll be glad to answer your questions.
Thanks, Professor Ayala, for your nice lecture. There is one question. No question. Professor Ayala, thanks. Professor Alex Brown, thanks again. Professor Mauro, thanks again for joining to us in this meeting. Rodrigo, do you want to talk?
Yes, I would like to say thanks for everyone. Enjoy us the second day, the condensement day. Then thanks again, Professor Alex, Professor Mauro, Professor Ayala. Bom.
Okay, thank you very much. Bye-bye.
Bye, Professor.
Okay, thank you. Bye-bye.
Thank you.
Thank you.
I also would like to say thank you for all the professors and for all the students, the chairs that were here with us along the entire day. And I'd like to say that we are finishing our activities for today. So we are going to go back tomorrow at 8:30 a.m. Brasilia time, okay? And I'm going to send the information for the chairs and the speakers of tomorrow of how access the Zoom as co-hosts in the end of this presentation. So thank you again very much for everybody and for the excellent contributions for our event.
Thank you. I'd like to stop the recording.